So, welcome back. So, in this class we will just continue those iron porphyrin system in catalases and peroxidase. So, still we are sitting on the heme enzymes. So, we have in our hand the only example what we are talking so much about the heme enzyme. So, they are also definitely heme enzymes, and now we will bring into the picture the involvement of H2O2. So, when it catalyzes the reaction of H2O2. So, these two reactions basically they are of similar nature. So, side by side we will talk together. So, you should clearly understand that one is catalase and another is peroxidase, and it goes sometimes through the same intermediate, where in the first case it is the reaction of one molecule of hydrogen peroxide with another molecule involving the catalytic activity of catalase, giving rise to water and dioxygen. So, this is basically our well known disproportionation reaction. This we all know. So, disproportionation reaction between one molecule of hydrogen peroxide with the other. So, one molecule of hydrogen peroxide is getting oxidized and another molecule of hydrogen peroxide is getting reduced, and at the same time we will see that some organic substrates or any other inorganic substrates as well can go for oxidation reaction using hydrogen peroxide. So, that is simple oxidation reaction. So, oxidation of a wide variety of organic and inorganic substrates, organic and inorganic substrates gives rise to a reaction like H2O2 plus our substrate is now AH2. So, is a oxidation reaction. So, this will be converted in presence of the catalyst, which is peroxidase will giving rise to 2H2O plus A, which is your oxidized form. AH2 is the reduced form and A is your oxidized form. So, large number of these molecules are well known to us, and well established forms of some of them we should know, because these are well studied. So, examples of one such is from the radish, so which is horseradish peroxidase. So, from the radish root we get this horseradish peroxidase. Then cytochrome C peroxidase is also well known. Cytochrome C peroxidase. Which is also abbreviated as CCP, and we get that from yeast. It's very commonly is available from yeast. Then another interesting biological molecule which we all know is a very good reducing agent is a glutathione. So, when glutathione is getting oxidized, so when your substrate is glutathione, so large number of these molecules are well known because they are operating on these substrates. So, when glutathione is getting oxidized by this peroxidase, we known as glutathione peroxidase. So, these peroxidase as well as catalase molecules, what we see that you have the same protoporphyrin 9, and that protoporphyrin backbone we can have. So, we have iron, then this is bound to this nitrogen of your porphyrin ring, so we all know now that is what are the myoglobin, hemoglobin and cytochromes. Then at one point it is forming your ferryle oxygen, that means all these cases it goes through this particular intermediate. So, this intermediate is there, and only difference is our binding for this position, that means the position number 5. If it is coming from a tyrosine residue, so if your tyrosine residue is directly using for coordination to iron site, we get a corresponding environment, because we know now through the protein crystallography and its corresponding X-ray structure gives us a clear picture and X-ray structure of one such species is beef liver catalase. So, beef liver catalase is there. So, you have, when we do not have this particular 5th position bound, it can interact with peroxides. Now, we know that this we are getting from hydrogen peroxide. So, you have the iron site, and iron site is nothing but your iron porphyrin site, and that iron porphyrin site can go for 2 types of reactions. So, one such environment is for catalase. So, for the other type of activity, that means the peroxidase activity, you can have a different environment. So, by looking at this particular position, that means this particular site, you can control the corresponding catalytic activity of iron in this particular environment, either for catalase or for peroxidase type. So, in this particular case, when you have separate isolated sites, and this for catalase, if we go for any such species which is iron, say in the trivalent state, which is bound to our porphyrin ring, so we get ferric porphyrin system. That means, if you have any ferric chloride in your hand in a test tube, and how it will be reacting with hydrogen peroxide, because those reactions with simple ferric ion is not so much exciting, but when it is attached to porphyrin, and this porphyrin when we put inside a protein structure, that means your other position, that means coordination number 5 is occupied by your tyrosine residue, then it basically controls the reactivity towards hydrogen peroxide. So, this particular binding will control whether it can react with hydrogen peroxide for a catalase activity or for a peroxidase activity. So, initially if we see, and people have identified, because already we have in our mind just now what we have seen in case of cytochrome C oxidase, that when a iron 3 based porphyrin system is there and which is reacting now instead of dioxygen molecule, because in case of cytochrome C oxidase we are talking about the binding of dioxygen molecule. That is why we are looking for the reduced form of iron site, but now the ferric form is there, it can very easily go for reacting your hydrogen peroxide system. So, it gives now Fe in +4 oxidation state, and now your site is oxidized for porphyrin, and you have this oxo. So, immediately if it gives this particular species, and which we already know that in terms of the corresponding redox equivalent, if we simply ignore the corresponding oxidation of the porphyrin site, and if you do not have the corresponding evidence for the oxidized ligand, because whenever you have a metal ligand system, you can either oxidize the metal site or you can oxidize the ligand site. But if we are unable to detect the corresponding oxidation of the ligand site like that of our porphyrin, we can say that no, porphyrin has not undergone any oxidation, it is your iron site which has been oxidized, but which is pretty difficult. But if we do not have any such evidence, we can write it as a iron in +5 oxidation state. So, that is very simple. So, iron oxo and the porphyrin plus water. So, this is a very simple and straight cut reaction. So, if you have your iron and it is bound to 4 nitrogen atoms of the porphyrin, okay. So, this is your entire porphyrin ring, and whatever thing is happening over there is due to your presence of X as well as Y. So, when it is reacting with say this hydrogen peroxide, so if this X is there and X is loosely bound, so X will go away. So, there will be removal of X, and at intermediate point it can react with O2. So, either the vacancy over here or any loosely bound water molecule or any other solvent molecule on this position can be replaced by your hydrogen peroxide, and it gives you some species which is based on your interaction with peroxide, and then it can go for your Fe oxo form. So, these changes now we know. So, these changes in your hand, and for this transformation what is forming immediately through the addition of hydrogen peroxide over here, we can label it as a compound which is compound 1. Why we are labeling it? Because we should characterize it nicely for its different solution properties. That means, what are its corresponding lambda max values, and immediately what we should know its corresponding color, and this color because there is a change, because all the porphyrins we know the basic ligand color is purple. When it goes for the complexation with iron, it changes one color, and then if you have hydrogen peroxide, then it changes to a green color, and it has some prominent lambda max values at like that of your cytochrome C oxidase at 655 and 405 nanometer. So long you are with this particular species, that means the oxidized porphyrin, and Fe2 plus Fe4, we get a corresponding signature for this particular absorption. So, we get compound 1. Then if we allow it for a corresponding 1 electron reduction, because this is the highest possible oxidation state in terms of either 5 or 4 with oxidized porphyrin. If you reduce it back, that reduction will give you the same iron in 4. What will happen to the porphyrin? Only the porphyrin is now your reduced one. So, 1 electron transfer in that particular case, either the porphyrin site or the iron site, is that between these two you can have a electron transfer, either it is on the porphyrin or it is from the iron side. But sometimes the covalence is that much that there is no such demarcation, that means the corresponding character, that means such ligand character or the metal character is mixed. It is very difficult to identify, that means where from the electron is coming out for the oxidation and where the electron is going for the corresponding reduction reaction. But when you allow it to react with hydrogen peroxide, then you can go for 1 electron reduction, immediately we get this species. So, this is our species of choice, that means your iron 4 porphyrin species, which is having a oxo bond, and this can be labeled as your compound 2, and it has a different color. So, the change in color immediately, without measuring the corresponding lambda max values, without knowing the corresponding spectral detail, you can tell that in this particular case this is one level of oxidation and this is another level of oxidation. So, in terms of the electron transfer, you can say that one is your green product, another is your red product, and this green and red product, because this red product has corresponding lambda max values of different magnitude, which is 568 nanometer one, then another is 536, and last one is at 529 nanometer. So, spectroscopically, so if you just apply UV visible spectra, so we will apply nothing, only UV visible spectra. So, UV visible spectra will tell us that you should be able to generate 2 different species, and these 2 different species when we have, and we next this can be allowed, because we are talking about for this catalase reaction, this is 1 hydrogen peroxide. So, we are having one more hydrogen peroxide, because one is getting oxidized and another is getting reduced. So, you have the ferryle iron in your hand, and that ferryle iron, when is allowed to react with another molecule of hydrogen peroxide. So, it is plus 4 P. So, it is compound 2, which we react with H2O2, we get compound 3. So, what is that compound 3? that compound would be we are utilizing our well known knowledge of hemoglobin and myoglobin is oxyform. So, iron is going down to Fe2 now and it is bound to dioxygen molecule. So, that is the way that not only forming this ferrile species, you can generate the dioxygen molecule there with the reaction of second molecule of hydrogen peroxide, that gives rise to the generation of compound 3, which is in reddish in color. So, which is not completely red, what is your compound 2, but which is reddish in color with a slight shift in the lambda max values from 568 to 585 nanometer, then 536 to 545 nanometer, and last one from 429 to 416 nanometer. So, all the 3 bands in the visible spectrum getting changed and color is changing from red to a reddish form, and that reddish form is responsible for the generation of compound 3, and this can also be formed because here with the generation of this O2 plus water, water will be produced from there, and this we all know now that this is very easily we can make from Fe2 porphyrin plus O2, because if you just take this in your hand and if you have a ferrous porphyrin, and that ferrous porphyrin when you allow it to oxygen, and we all know that depending upon the corresponding partial pressure and the medium everything, the corresponding oxyform you can generate, and this oxyform if you can generate, which can easily be characterized by knowing the corresponding lambda max values. So, simple characterization in terms of these lambda values will tell us whether you have the compound 1 or compound 2 or compound 3, because in one particular case same iron center is utilized for the oxidation reaction, in another case it is utilized for the reduction reaction. But during that transformation something is happening, that means when you start from one particular step, that means ferric porphyrin we are starting and generating the ferrile iron center through the use of 1 hydrogen peroxide, and that highly oxidized form of this ferrile iron. This ferrile iron is now what? Ferrile iron is your stronger oxidizing agent now compared to hydrogen peroxide. Because of this nature, that means it is a stronger oxidizing agent than hydrogen peroxide. Earlier we are utilizing hydrogen peroxide for oxidizing your iron side. Now your ferrile center is more potent oxidizing agent, which is utilized for the oxidation of hydrogen peroxide. So, you see that iron side we are simply just changing from one side to another, that means your environment is responsible as well as the binding of the form and how easily you are able to produce the corresponding ferrile species by manipulating the corresponding E0 values. So, we get the different properties. So, how these different properties we are getting, because this is one example for our catalase compound, that we will see in our next example for peroxidase, that different properties which we all should keep in our mind, because we are starting our discussion from cytochrome C oxidase, because one after another we have to compare, because all the sites at one point of time when you know all these things, you should be able to compare that why it is showing a reactivity related to cytochrome C oxidase, why it is peroxidase, why it is catalase, and why it is some other molecule which is will be cytochrome C450, that we will see our next molecule. So, different properties are originating from different electron densities, which is important. So, different electron densities where at the metal site. Different electron densities at the iron site, which is very important. Plus, we can have something, that means we are changing from one protein environment to the other, because the for catalase the protein environment is different compared to your peroxidase, which is also different for your cytochrome C oxidase. So, the second sphere of environment, that means you have the corresponding electrostatic effects of the protein, that means the different of the different protein environments. So, this particular one, when we get that how this protein environment is changing, that we will see that when we have this particular catalase activity and when the site is loosely bound, that means if we just see little bit related to the 3-dimensional structure of our simple porphyrin ring. So, is a very four porphyrin rings, sorry, 4 pyrrole rings should be visible, and this iron site. So, we have one nitrogen, this nitrogen and this nitrogen and this nitrogen. So, when this is the vacant site, you get a corresponding bowl like structure. So, you just if you just see, it is visible from this side. And you have a typical bowl shaped structure, and iron is sitting over there. So, this particular site, which is your apical site, because all 4 basal sites are occupied, all 4 basal sites around iron are occupied. So, within this particular one, and it is again your the same example, the Biff-Lieffer catalase. Why it is bowl shape? Because you have the bonding from the tyrosine residue. Its numbering is 357. So, this particular one. So, when you get this, that means you have this particular arrangement and the protein environment is also supporting this arrangement. So, this bowl shape structure, so bowl is basically attracting those substrates. So, whatever substrates we are getting, so this is getting attracted over here. So, different substrates will come and will just go inside the bowl, and the corresponding reactions we see. So, people are trying basically. Why we are talking so much about all this? Because we know that you have the corresponding accumulation of H2O2. So, on all living organisms, including human being, including man, you have tremendous accumulation for this hydrogen peroxide, because we are consuming our dioxygen for regular survival. So, when we put dioxygen into the system for our glucose oxidation or food oxidation or anything, we all the time, and we know that some amount of superoxide is forming, then some hydrogen peroxide is also forming. So, there should be some suitable mechanism to destroy this. So, we are able to destroy some of these hydrogen peroxide molecules through the mechanism of this catalase activity, that one hydrogen peroxide is reacting with another hydrogen peroxide through this iron site. But at the same time, if we can have some arrangement, that means you have FeO2 2 minus, that ferryle has not formed, iron site is there, that means you have activated hydrogen peroxide. So, your hydrogen peroxide, because we all know that various reagents, various reactive species sometimes getting activated with the use of our metal site. So, it is iron site activated hydrogen peroxide. So, that can also be a very good reagent for destruction of some of other toxin molecules. So, some of other toxic molecules, that means different toxins. If you consider that hydrogen peroxide is also toxic for our body, so accumulation of hydrogen peroxide is not good for our health, for any living being. So, that we are able to destroy, that means the concentration of catalase in all living system is important. That means some person is there which are unable to produce the amount required for the destruction of hydrogen peroxide, and some others living species is producing more hydrogen peroxide. So, there will be some imbalance. So, this particular reactivity can be useful also for destroying some other toxins like formaldehyde also. That is why we are studying this. That means, what information we are getting from this living system, that catalase we know, and catalase is going through that mechanism. So, definitely if we are able to use that for destruction of hydrogen peroxide, we should be able to use that system for the destruction of formaldehyde. We can use that for the destruction of formic acid also. Sometime we can use this particular arrangement for destruction of phenols also and alcohols. So, the different for different species, that means when you use that catalase activity, we have the iron porphyrin site, and when we go for the corresponding peroxidase, and the solution study is also very simple, that we should go for knowing the corresponding particular species, because the peroxidase are also available everywhere starting from plants. So, the plant origin is basically our HRP, the horseradish peroxidase, and which is available from the roots of the horseradish. Then the peroxidases are available in animals also. It is also available in bacteria, and these are very small molecule species, and this iron 3 heme unit in peroxidases that means iron 3 porphyrin unit is monomeric, having only molecular weight of 44,000. So, here also we have iron site which is bound to 4 nitrogen atom of the porphyrin ring, and in case of catalase, as we have seen that this particular position can be occupied by some water molecules also, and this water molecule has to be stabilized. If it is bound to water, it is not that the only water molecule like our hexaco species, hexaco metal salts. That means, this there will be a pocket, and hydrophilic pocket if you are able to form, and that bound water molecule which is supported by weak coordination from this iron, and this apical site binding is little bit weaker all the time because you have a iron oxygen long bond compared to iron nitrogen bond, and this is hydrogen bonded with some side chain of the protein chain which is histidine. So, if you have a nearby protein chain, say this protein chain is continued with aspartate, then cysteine sulfur, cysteine group, and then phenylalanine and so on, and histidine, then this side also phenylalanine and this side. So, very useful hydrogen bonding interaction can be provided by this histidine unit. So, this is all the time very important, this interaction, not only knowing the iron site which is bound to so other groups, because this histidine binding is important. We will see, because this site we are utilizing not only for water binding, but afterwards what we are seeing that this we are utilized for binding of O2, binding of hydrogen peroxide, that means sometimes hydroperoxide HO2 minus. So, they are all related, how oxygen is bound to your iron site, how hydroperoxide is bound to our iron site. So, if this particular species is getting stabilized, and if we have some vacancy over here, we will find that this water position can be occupied by your hydroperoxide also. So, that we actually looking for, that binding of hydrogen peroxide or hydroperoxide to the iron site, followed by hydrogen bonding interaction with the histidine group, will also lead to some amount of reactivity pattern, where we will find that there will be, instead of this water molecule, there will be OO bond if it is hydroperoxide. So, that OO bond which is terminally stabilized by histidine residue, and that we all know the charge separation will be there, delta plus and delta minus end will be stabilized by your histidine unit, then there will be a possibility for your OO cleavage. So, this hydrogen bond interaction will be useful for our OO bond cleavage. So, this particular case, if you have on the lower end you have the histidine unit and then other subunits like glycine, like threonine, etc. So, this particular case we just determine that if we have iron site, and this iron site like catalase will find that you have compound 1, we have compound 2 as well as compound 3, and these particular cases we just can compare by knowing their corresponding different lambda max values, and those lambda max values will also immediately tell us when we talk in terms of the corresponding EPR measurements, that what type of iron site you have. And this particular case, if we have a high spin iron 3 which is very much EPR active, for its S is equal to 5/2 value, and this particular case immediately we see, since it is high spin, that means there is some interaction. That means it is not that all the time when it is bound to your water molecule or it is bound to your histidine. So, it is this histidine is your different histidine, which we also known as distal histidine. And this coordinated histidine also plays some important role to give you a arrangement for iron site, whether it is high spin or low spin. So, this particular site, depending upon its corresponding pK value, that means this nitrogen will have a typical pK value. So, if we just monitor the pH, and the same example of this your horseradish peroxidase HRP sample, that you have two different pH values, one is low, another is high. So, one is low pH and another is high pH, and low pH, that means your all these sites, the nearby sites are all protonated. Your histidine site is protonated. So, you do not have anything, and most of the cases like your catalase site is also when you have the phenol coordination. So, that phenol can either be unprotonated or protonated depending upon your pH of the medium. So, when it is low, you have the protonated form of these things, that means some of these nitrogen groups are protonated, that means you have weak field. So, you will get a corresponding high spin state, and in case of high pH where it is basic in nature, you get a low spin state. So, when we talk about this interaction in related to your spin state, we should also be careful about the different spin states around the iron, and this particular spin state will have different reactivity pattern with that of our water molecule as well as hydrogen peroxide. As we all know, ultimately it is also related to the binding of dioxygen, because how your deoxy form of hemoglobin and myoglobin, in which form, whether it is in the low spin state or in the high spin state, it is used for binding the oxygen molecule, that we know for our knowledge from your myoglobin and hemoglobin. So, these two spin states are important for binding, and your hydrogen bonding interaction from the distal histidine is also important. So, now we can see that this particular form, when it is giving us the corresponding coordination, that means you have now nitrogen from the imidazole unit, and these are the porphyrin groups. So, like catalase, it also forms a corresponding ferryle unit. So, now you know that the electronic parameter around iron is different, because in case of catalase it was your phenol unit. So, now the imidazole side chain, imidazole group at the coordination number 5 plays some important role for a different type of reactivity for your peroxidase activity, because this peroxidase activity is responsible for your oxidation of the substrate. That means, you have H2O2 as well as the substrate. In case of catalase, we are talking about two molecules of hydrogen peroxide. Now, you have the substrate as well as the hydrogen peroxide. So, if you have a typical substrate for this activity of the peroxidase, your substrate should also be nearby. That means, you are talking about something that you transfer this oxygen to this unit, or you just simply oxidize it to A. So, your substrate should also be close to the catalytic site, and the generation of this is very important to know that if you have this iron site which is in the trivalent state bound to nitrogen, and this is another nitrogen, and when it is simply reacting with hydroperoxide, that means we have used hydrogen peroxide for the reaction, but following a deprotonation it can go for binding with OOH, because this is looking for a charged coordination and iron is in the +3 oxidation state. When we talk about +2 oxidation state, we can handle about the dioxygen molecule which is a neutral one, but when it is a charged one we have to bind this one for this particular type of arrangement. So, here you have a charge separation of delta plus and delta minus, and this one, therefore, this oxygen can have hydrogen bonding interaction as well as this will also have hydrogen bonding interaction with the hydrogen of the corresponding nitrogen of the distal histidine. This is the binding. So, this nitrogen, the second nitrogen, this NH nitrogen is involved in hydrogen bonding, but the second nitrogen is also sometimes depending upon your pH of the medium can be protonated. So, this hydrogen can supply a proton to the system, can also accept proton from the system. So, this particular unit, so it is very close to your iron side, and you see we are having some extra proton from this, because we are talking about here that HO2 minus plus H plus. So, this proton can go and bind and sit over here. So, these particular arrangements for the stabilization of this arrangement as well as the other end, that means the positive end, can give rise to something which is coming from positively charged other amino acid end. So, is arginase. If is a positively charged arginase site is available and which is close to this oxygen, so this arrangement is stabilized within the protein pocket. So, that is why whenever we determine some excess structure of this protein molecule, so this biomolecule, when we determine this structure, always we talk about the FeO angle. Not only the O distance, which is also important for its knowing the corresponding bond order, that whether it is a single bond or a bond order of 1.5 or a double bond character, and how easily it can be clipped, how easily we can go for breaking this OO bond. So, that will be known by knowing the corresponding OO bond, and at the same time FeOO bond angle also. You see, so the hydrogen bonding push from this side and another interaction from the arginase side will stabilize only this arrangement. That means, at a particular angle it will be stabilized, this particular arrangement will be stabilized. So, when this weak interaction is taking place and we are getting this particular arrangement, so in the second step what will happen, we should immediately able to write this, that you have Fe and this particular one will go for making FeO, that means the ferryle system we are getting. This is for the nitrogen of the imidazole ring. So, these two nitrogens are there, and what will happen to this part? This part will go like this nitrogen, and you have this is also remaining, this is going there. So, you have HOH and positive charge arginase. So, the cycle continues. So, the immediate movement for this, that means you have this we all know, that means your NH. This NH is important. So, this proton will now move to this nitrogen to regain this structure. So, you see in this particular case, that not only this particular arrangement, that means the coordination environment around iron is important, but also the secondary interaction, the second sphere of interaction, which we call the protein environment. So, here also this protein environment is important. How protein environment plays an important role, that is known for from here. So, protein environment, this basically controls the typical arrangement of this O2H unit, and which can also control the OO heterolytic cleavage. Because the terminal end is attached not only through hydrogen bonding to the imidazole unit, but also some arginate side chain. So, this arrangement then immediately gives us some idea that we can see some of these reactions, because some of these reactions are very useful for that oxidation reaction. So, whether we can utilize these simple informations related to our catalase and peroxidase system for oxidation using some model systems. It is very important to design, is very important to make all these things. That means, if you have some substrate like alcohol or some aromatic compound like phenol, then we can have the H which can be oxidized using your hydrogen peroxide using some iron compound, iron 3 compound, and model compound means we have some laboratory prepared compound. So, one such binding model is that if you have a catechol type of arrangement, so that catechol type of arrangement can be utilized, giving you a H, that means this is there, that means if you have a benzene unit like C6H6, you get a corresponding phenol plus H2. So, what we are talking therefore, basically the next system what we will talk in our next class is the corresponding hydroxylation reaction using some related molecule which is cytochrome C 450, cytochrome C, based P 450. So, here in this particular case, when you have a bidentate chelation around iron, we have Fe which is Fe3 plus this binding is there, So, this definitely react with this particular one in this form. That means, this interaction, whether you have the Fenton type of arrangement, that means iron, that means it is attached to your ligand system. So, the ligand will play some important role to know all these reactions, and then you have the hydrogen peroxide, and if this particular unit is there, that if it is a catechol, which is a very useful ligand. So, O is nothing else but your catechol ligand. So, that catechol is there, and that particular one ultimately can give rise to some of the oxidation reaction to produce quinone. Sorry. Quinone system, and this quinone at the end basically can weakly bound to your Fe2 plus system and O2 2 minus. So, this particular picture tells us so many things, that if you have the ligand, and if your ligand is functioning as your substrate AH. So, if your ligand catechol itself is a substrate, so your substrate can be oxidized. Substrate can be oxidized, and at the same time this particular one, whether it is loosely bound to hydrogen peroxide, and then you have this OO bond cleavage, that means some interaction you can have where you have the FeO interaction, it can be your Fe double bond O also. That means you have something in your hand, that electron transfer can take place, and electron transfer can give rise to the corresponding oxidation of the substrate from catechol to quinone, will end up with the quinone system, or you will should be able to generate a reagent where you have oxo function is sitting on the iron, because not only any organic substrate, but you can utilize iodide, you can utilize Br, you can utilize thiocyanate to get the corresponding hypoiodide species, hypobromide species, or hypothiocyanate species OSCN minus. That means, it would be a very useful technique to know that how we can activate the hydrogen peroxide, and during that transformation whether your oxidation equivalent, that means your oxidation equivalent is residing on the hydrogen peroxide part, and that hydrogen peroxide part is moving from this hydrogen peroxide to O2. If it moves from O2, its corresponding reduction equivalent is transferred from this side to other side. But if this, you can consider this as the reagent and this as the substrate, your mediator is your metal center. So, always in studying all these systems, that means the different model system, starting from our cytochrome C oxidase, catalase, peroxidase, all these things, the first thing what we have identified is that your iron site, that means which is your metal based catalytic site, and you have the other part. When you have the porphyrin unit, we have seen, or we have the corresponding tyrosine unit, which was Y. So, we were at one point we were able to oxidize it to this or the corresponding starred version of the tyrosine. So, through this oxidation process we are able to generate some organic radical. So, this particular case, this generation of these two organic radicals are important for different iron based reactions related to hydrogen peroxide and its oxidized form. So, next day we will see something related to your cytochrome P450. Thank you.